This is the podcast to help you get ready for the Chapter 12 test that's coming up this week over genetics. First thing we'll start out with is the vocab words. First word is heredity, which is the passing of traits from parents to offspring. The next one is trait, which is a specific characteristic that an organism can pass to its offspring through its genes. Followed by purebred, which is an offspring of many generations that have the same trait or the same form of a trait. The next word is gene, which is a sequence of DNA that determines a trait and it is passed from parent to offspring, followed by alleles, which are the different forms of the genes. Then hybrid, which is an offspring of crosses that have two different alleles for a trait. We have phenotypes, which is an organism's physical appearance or the visible traits, and to go along with that we also have genotype, which is an organism's genetic makeup or allele combinations. There's homozygous, which means that they have two identical alleles for a particular gene, or there's heterozygous, which means they have two different alleles for a particular gene. So homozygous are for identical, heterozygous are for different. As far as dominance goes, we have incomplete dominance, which is a situation in which one allele is not completely dominant over another. And then we have codominance, which is the situation in which both alleles for a gene are expressed equally. Then we have meiosis, which is the process that occurs in the formation of sex cells, which are the sperm and the egg, by which the number of chromosomes is reduced by half. And then clone, which is an organism that is genetically identical to the organism from which it was produced. And then finally, inbreeding, a selective breeding method in which two individuals with similar sets of alleles are crossed. The next item are the different concepts. We'll go through first what the process of meiosis does, follow that by the difference between asexual and sexual reproduction, then move on to how genes, chromosomes, and DNA are related and what exactly they do, Punnett squares and how to use them and when to use them, and then finally some genetic problems that relate to the probability with Punnett squares. So first of all, what does the process of meiosis do? So it's a process of making new sex cells, which are of course the sperm and the egg. Sex cells are made up of exactly half of the total number of chromosomes that are typically found in a cell. If we take a look at the process here, you can actually see it starts out as you have a regular cell that then undergoes what looks like the process of mitosis, and it actually makes a copy of all the DNA or all of the chromosomes or the DNA and creates two separate cells, which are called daughter cells. Then what happens at that point is those cells actually then go through another split, but they don't make a copy before they do that split, which gives you your four cells coming out of the original one cell, and it also means that they each have exactly half of the original number of chromosomes, which allows them to undergo the process of sexual reproduction then, because you can get the variations that occur. And that relates then to what is the difference between asexual and sexual reproduction. If we take a look at the box and T-chart that I have here, you can see that both of them create new organisms. But there's a lot of other differences of the fine details that go on. For example, comparing asexual to sexual reproduction, we can see that for asexual they're identical to the parent, whereas for sexual they're different from the parent. We can also see that asexual requires only one, while sexual requires two. One's faster than the other. And the big difference that comes into play, especially when we talk about the ideas of natural selection, and then also with the idea of how the genetics work and the different genetic issues that we can have, is that asexual produces the exact same thing. It's essentially a clone of the parent because it has the same DNA as the parent. Whereas when it undergoes sexual reproduction, you actually get different DNA that's a combination of the two parents. And of course then we have the process of mitosis versus meiosis that occurs. Now of course asexual reproduction does occur for cells replicating in our bodies, but as far as actually creating new offspring, it requires sexual reproduction. So when we take a look at this then, how exactly do these genes, chromosomes, and DNA, and what are they actually doing and how are they related to each other? Of course we know that two chromosomes form a pair. That's why we have 23 pairs of chromosomes, 22 that are both X, and then we have the final sex chromosomes, which are the 23rd pair. Each pair has one chromosome from each parent in the case of sexual reproduction, and each one of those chromosomes are made out of genes, which are found on there, and they're the smaller parts of the chromosomes. And of course, the chemical name based on this is the DNA. Now, the genes are passed from parents to offspring via the chromosomes. So that's what the purpose of the chromosome is. It actually transfers that information from the parents to the offspring in order to encode what the offspring's appearance and other characteristics will be. And this is all, as I said, comes from the DNA, which is what they're actually made of. It's deoxyribonucleic acid is actually the official name for it. So now we need to talk about Punnett squares, and we spent a ton of time working on Punnett squares and looking at how to use them and when to use them. Anytime we're looking at sexual reproduction, we can always use a Punnett square to help solve the problem. It makes it much easier than sitting there and trying to do like drawing the marbles out of the bag or something like that, and we can predict the probabilities that different things will occur. So if we take a look here, I've drawn a very simple Punnett square, just picking two arbitrary letters B and Y. And you can see I have homozygous on both situations here. So if you look at my mouse here, I'm circling right now, for one parent, it's homozygous on the B allele, whereas we have homozygous on the Y allele for the other parent. And you can see in this case, these are the four possible combinations that we can get from these two parents. In this case, they're all going to be heterozygous, so we have exactly four chances that come out at 100% being for the heterozygous situation. But it varies depending on what kind of what the parents have and exactly which alleles are going to be expressed in which, and we'll take a look at each one of those situations. Now, it actually makes a little more sense to talk about probability next, so that's why we're going to move into probability next. And we'll look at the probability with Punnett squares. And what's really great about a Punnett square is that you can just look at the number of boxes or cells that have some characteristic and then just say that 25% is for each box in this case. Now obviously if you go to like a double Punnett square or something like that, the probabilities become a little more complicated. But again though, it's a little bit easier mathematically because you can just figure out how many there are and divide it by 100, or divide 100 by that, and you can figure out roughly what the percentage will be for each one of the different boxes. And obviously if you have two boxes the same, you need to add, or you need to multiply that 25% by 2 to find out that it's 50% in that situation. So let's move on then to some genetic problems. There's actually four major genetic problems that we talked about in class, and we did several investigations on all of them. First of all, there's the idea of codominance, and then incomplete dominance, followed by complete dominance, which is where the idea of dominant and recessive alleles come in, and then sex-linked genetics or sex-linked traits. So first of all, we take a look at codominance. This is a situation in which both alleles for a gene are expressed equally. And so we can see in this case that both are in uppercase, because we always denote the dominant allele as uppercase and the recessive as lowercase. But when we're talking about codominance, they're both going to be of equal level of dominance. And so what we can see here is I've actually picked out the situation of looking at the red and the blue chips that we use. And you can see that I have a red parent and a blue parent, and we know then that all their offspring will of course be purple in this case, because you can see that when we cross the reds with the blues, we always get them written out as being, or we get all four of them being identical with the same phenotype, which is their appearance being purple, and then their genotype all being the heterozygous of the red and the blue, giving us a 100% heterozygous. And that's just one situation. Obviously, if you switch these over to this one to R and B, you would get something slightly different. I'm not going to go through that in detail because you guys can manage to do that easily on the test, which I recommend you do every time you come to one of these problems that you're trying to figure out. Just draw a quick Punnett square and work on it. It'll make your life much easier. Now we'll take a look at the idea of incomplete dominance. This is a situation in which one allele is not completely dominant over another allele. And so this is a kind of a interesting situation where we can actually get both expressed at the same time. Alright, so an example we saw in the book was the brown and the white chicken, or the black and white chicken. You can actually see on here that if you take, when you cross them, you can actually get some different combinations. Now in this case I decided to take a homozygous white chicken and cross it with a brown and white spotted heterozygous chicken. And so you can see that one parent has brown and white, both alleles present, whereas on the other parent they are homozygous on the white chromosome, or on the white alleles. And so we can see then that the different combinations we actually get two that are homozygous, and then we get two that are heterozygous. And so if we look at the phenotypes, again we have 50% that are white and 50% that are both brown and white. In that case they're actually more like a spotted or a speckled appearance. Then for the genotypes, we can actually get the white. We can see then that the genotypes still break down, though, that we still have 3 genotypes and 3 phenotypes. And that's really important to notice. We can't have more phenotypes than we do genotypes because we can't have any more ways to encode it. Now, if we take a look at complete dominance, on the other hand, we can see that one allele is dominant over another and will be expressed over the other. And this is where we actually have the upper and lowercase come into play again. And we can see that the uppercase one, for example, in this case is blue, and then we have red being the lowercase, so red is recessive, blue is dominant. And we can actually see that as we go through and do this cross, we have I did two heterozygous, and so we have two blue parents, and they can actually have, in this case, a red offspring. Now, when we take a look at the phenotypes, we can see there's only two phenotypes here. Because remember, since this is dominant, the BR is actually expressed, or we actually view it as being blue because the red is a recessive trait. And so if we're a parent to have, in this situation, for a parent to have the red offspring, they would actually need to have both of the alleles being red for one of their offspring. And they have a 1 in 4 chance or a 25% chance of that occurring. So you can see here also we have 3 combinations for genotypes but only 2 phenotypes, and it all has to come down to the idea that we have dominant and recessive trait, or dominant and recessive traits, or dominant and recessive alleles. The last one we looked at was the idea of sex-linked traits, which these are traits and alleles that are found on the X chromosome of the 23rd pair, which is also known as the sex chromosome. And so in this situation, it actually gets a little more complicated for some of the math that we have to do, because we have to also now start dealing with the idea we have males and females, and we have some kind of a trait going on here. And so when we look at this situation, we're going to be looking at the idea of male pattern baldness, which we saw follows the female lineage, so you can actually see that it follows on the X chromosome. And of course, on men, they only have one X chromosome, and then they have the Y chromosome in their 23rd pair, whereas for females, we can actually see that they have 2 X chromosomes. So in this situation, we can see that we have a non-bald mother and a non-bald father, because we're saying that baldness is the recessive trait, so therefore it's the lowercase b, whereas not being bald is the uppercase b. And so when we look at this situation here, the offspring, you're going to, no matter what, not have a bald female. So it's 100% non-bald female, but you can see we have 2 genotypes. So while we have all the non-bald females, it's a potential possibility that non-bald female, or one of them, could have a bald offspring later on when we look at the F2 or the F3 generation. And we look at in the case of, for the males, since they only have one X chromosome, the recessive trait will be expressed in that situation. And so we can see right here that the recessive trait is being expressed for this situation because we have a bald male that appears. Now when you look at my probabilities over here for the phenotypes, you can see that I said there's 50% bald males. And there are 50% bald males because I'm saying that it's both bald and male. Or I'm saying that I already know it's going to be a male, and so therefore if it's bald, it will be a 50% chance. The mathematics can kind of go back and forth depending on how you're actually going to describe this, but as long as you make it obvious, either way it will work. So finally, just a couple other things just to remind you of. Make sure you study your claims and notes from the investigations in this chapter. There's a lot of probability problems on this test, so make sure you look over those and you're ready for them. Remember, there's really only like 6 or 7 situations that we saw that really can show up. So as long as you understand how to apply those situations, then it'll make it very, very easy for you. Make sure you also study the vocabulary with a friend or a parent. Remember, all the vocab words we actually wrote down can and will be used in the test. Just the ones that I listed are the ones that are a little more likely to come up. And remember, as I said, there are application questions on this test. The short answer and also the multiple choice are application driven in this test. If you have any questions, you can jump onto My Big Campus and ask them on there, or you can ask them during class. Good luck.